Once upon a time, as all good stories begin, there lived a cell line. The M14 cell line comes from a 33-year-old man who died of melanoma in 1974. Just like a tree, this young man's melanoma cells continue to grow, giving a lineage used for decades of cancer research. But M14 is only one lineage tree in a vast forest of cell lines. More than 120,000 cell lines are known to exist in laboratories worldwide. A closer look at this forest of cell lines reveals many trees that grow from incorrect material. Misidentified cell lines weaken our research and slow down new discoveries. A cell line can become misidentified through cross-contamination. It only takes a single cell to pass from one dish to another, maybe in a single drop of liquid. If the new arrival comes from a cancer cell line like M14, it usually has a growth advantage. Rapidly growing cancer cells outgrow the original cells and eventually replace them in the culture dish. Over 20 cell lines overgrown by M14 are used as models for unrelated diseases such as breast cancer. Cell lines can also become contaminated with microorganisms. Bacteria and fungi may be obvious when you look at the cell line under a microscope, but other microbial contaminants may be hidden from view. Mycoplasma are tiny bacteria that stick to the cell membrane and can enter the cell to infect it directly. Viruses can multiply inside the cell until they are ready to emerge and infect other cells. Cell lines are living organisms that can adapt to suit their environment. Cell populations with different characteristics can coexist in the same culture dish. Changes in growth conditions select for some cell populations at the expense of others. Over time, this leads to evolution in the behavior of the cells and in their chromosomal content. Any cell line can become contaminated or misidentified, and any cell line can evolve in culture. Cell line problems can be avoided through good cell culture practice. Look for a reputable source that banks the cell line early and tests it for contamination. Preserve each cell line by preparing frozen stocks before you start to work with it. Authenticate your cell lines using STR profiling, SNP analysis, or other genotype-based techniques. Use good aseptic technique and test regularly for mycoplasma and other hidden microorganisms. When you publish, provide sufficient information for other people to replicate your cell culture work. And above all, never take your cell lines for granted.